All right, so we're gonna get started here with an afternoon keynote talk by Dr. Brian Fallon, director of the Lyme and Tick-Borne Disease Research Center. Again, a friend from across the Central Park here in Columbia. I had met Brian before this conference, and I knew I was going to be introducing him, so I went to do some background and talk to people that knew him. And of course, his publications are outstanding and speak to a lot of the work that he's done on the science side, but I also had talked to a lot of people that, or at least knew people that had seen Dr. Fallon as a patient. And I think what impressed me almost more than his publications was how enthusiastic and how grateful the people were that were seen by him as a physician, as a doctor. So it's rare that you get someone that is both a prolific scientist and someone that has a big impact as a clinician and taking care of patients. So that was super impressive to me. With that introduction, I'll welcome Dr. Fallon to the stage. Thanks.
So I was thinking as I was sitting here earlier that it's quite amazing to have a meeting on omics and precision medicine for Lyme disease. Never would I have thought, like 5 years ago, that this would have been possible, that there would have been so much openness to the possibility of persistent infection, that there would be so much openness to the problem of chronic symptoms after Lyme disease. So it's a delight to be here, and in 25 minutes I'm going to review the entire history of Lyme disease, probably in about 3 or 4 slides, and then talk a little bit about what we do at the Lyme Center and finish up. Okay, so a few early pioneer researchers and clinicians. So Willy Bergdorfer, we all recognize, Jorge Benach, and Alan Barber. Those were the 3 key people who were at Rocky Mountain. Willy and Alan were at Rocky Mountain Labs. Jorge Benach was working on Long Island collecting ticks. And very interestingly, he was collecting ticks because he was studying Babesia, but he was also interested in this new disease called Lyme disease. So he was storing serum for study. And when Willy was dissecting ticks that Jorge had sent him from Long Island and found these spirochetes, he immediately called Jorge Benach and said, "Do you have any serum on patients with Lyme disease? Because I think this might be the cause of Lyme disease." And because he had that repository of samples, Willy and Alan were able to then determine that this, in fact, was the cause of Lyme disease. So the key message from this slide is that repositories are extremely valuable. The other interesting thing was that some of the patients with babesiosis had reported a pre-existing rash. And so he was also able to document that those patients with babesiosis also had Lyme disease previously. So that was one of the first stories of human co-infection with both of them. One of the things Alan did early on, Alan Steer, was created a serum repository, and that formed the basis of the CDC repository for many years. That's been replenished and renewed since, but that was a very important contribution by itself. He identified Lyme arthritis, of course, clarified the role of autoimmunity, identified genetic risk with HLA-DR4, and what I really like about some of the papers that Alan has written is that he's been very honest about what he sees. And he reported in the New England Journal of Medicine something that was very important, that 20% of patients with infection may be missed because they don't demonstrate the objective signs. And many clinicians will refuse to make a diagnosis of Lyme disease unless they see those objective signs. And here in the New England Journal of Medicine, Alan is saying, don't miss those patients. He also, of course, reported that antibiotics may fail, causing refractory Lyme arthritis, but also in a small group of patients with intracranial pressure that in one case led to blindness in a child. So he reported the bad outcomes as well as the good outcomes. And recently there was a proteomics study that he conducted that led to the identification of the first autoantigen in refractory Lyme arthritis, endothelial cell growth factor. So these are all major contributions by key people. But not to be forgotten are the clinicians who have been struggling in the community. And early on, these were the clinicians who were focused on the outliers. These were the clinicians who were focusing on people who weren't getting better with the standard courses of treatment, or who were relapsing, or who didn't present with the typical signs. And these people often were castigated by some of the academic experts. So Dr. Joe Burriscano created some of the early diagnostic guidelines for people with chronic symptoms. Dr. Ken Ligner also treated a lot of these patients with chronic symptoms, but to his, one of the things he did to his great credit was to collect serum and spinal fluid samples on all of his patients that he saw, stored it in a freezer, so that others could study it in the future. He still has that freezer waiting for people to study it. So, and he has enormously detailed clinical documentation on his patients. And he collaborated with scientists. So he invited the CDC to collaborate with him. He sent samples to them, and one of his patients actually was culture positive in the spinal fluid, despite prior antibiotic treatment for Lyme disease. Ed Masters was another clinician. He was from Missouri. And in Missouri, there were no Ixodes ticks, and so it was extremely impossible that people had Lyme disease. But he was seeing these rashes that looked exactly like Lyme rashes, and it drove him crazy because he was saying it's Lyme disease, the CDC was saying it wasn't. So rather than just fight with each other, they collaborated and learned a lot with Gary Wormser as well, and learned a lot about star eye, otherwise known as Master's disease. So when I look at the history of Lyme disease, this is a very personal history. I sort of divide it up into three segments. 1976 to 1990, which I think was a time of great discovery and openness when they were first describing the clinical manifestations of Lyme disease, first trying to figure out how to treat it, finding the Borrelia spirochete as the cause of Lyme disease. And then in 1990, the Public Health Department of the U.S. realized that we needed to really understand whether it was spreading, how much was spreading, how much the incidence was increasing. And so they create these narrow objective markers or signs of disease, as well as they identified lab criteria for diagnosis that were very highly specific. And this was done for very important epidemiologic purposes, which was to objectively monitor the change of disease over time. But clinicians sometimes were using these surveillance criteria inappropriately as the sole means of diagnosis, and therefore some patients weren't getting diagnosed. So that created an enormous amount of hostility in the patient community because many patients weren't able to get treated or cared for when they felt that they had Lyme disease. It created conflicts because the insurance companies were no longer willing to pay for treatments that didn't meet for cases that didn't meet the CDC surveillance guidelines. So it became a very hostile and unpleasant period in the Lyme disease history. Then in 2008, there was this time of renewed discovery and exploration. So I'm going to go to the next slide so you can see this a little more visually. So childhood arthritis, 1977, spirochetes in the tick, 1982, Lyme, the new great imitator. That was Andy Packner's paper, in which he described some of the atypical manifestations of Lyme disease, including a child who developed new onset OCD after Lyme arthritis that didn't respond to standard psychiatric treatment, but did respond to antibiotic therapy, or a case of an MS-like illness that responded to antibiotic therapy. And then came the surveillance, CDC two-tier criteria. The Borrelia genome was sequenced in 1998. That was very important, of course. That led to a lot of where we are today. The vaccine came out in 1998, but it only lasted on the market for 4 years because for a variety of reasons. One was that it wasn't as effective as people hoped for all people. And secondly, there was concern about possible side effects from it. And then there were the clinical trials. And sometimes we think that the NIH has ignored the problem of chronic symptoms. But in fact, they actually funded 3 clinical trials. One was the one we did, the last one that was published. But there was one Dr. Klempner did, published in the New England Journal of Medicine, that had negative results. Lauren Krupp's study at Stony Brook showed that patients did in fact respond with improvement in fatigue after 1 month of repeated antibiotic therapy, intravenous ceftriaxone, but didn't improve on some other measures. But there's been a great deal of ambivalence about how to interpret these trials. Some people have said, if you read the guidelines, it says the trials have been uniformly negative. If you listen to me, I say that they weren't uniformly negative. If you listen to others, they say they weren't uniformly negative. So how can something as objective as science have so many different interpretations? And partly it's because these are just small studies, and partly it's because there's only been 3 studies. So one thing that Dr. Zhang said before, which I certainly agree with, which is that we do need more clinical studies of the treatment of Lyme disease, especially those with persistent symptoms, to see what benefits them. And then, of course, we have the paper that came out in 2008, which was Steve Bartol's paper on the persistence of Borrelia in the mouse model. And that was a game changer, I think, in our community, because basically when someone of his preeminence comes out and says that there is persistent Borrelia after treatment in the mouse model, all of a sudden people start paying attention. And then Monica Embers and Mario Philippe published their paper on the monkey model. Other mouse model studies had similar results in Finland as well. But if you go back to 1995, you see a paper in the Journal of Spirochetal Disease that never made it to PubMed. But Reinhold Straubinger in 1995 reported persistent Borrelia after antibiotic therapy. So many of the clinicians who went to those conferences sponsored by the Lyme Disease Foundation knew his work, but the rest of the academic community actually didn't. And much of this work, and in terms of education, in terms of funding of research, wouldn't have happened without angry mothers who were fed up with the nonsense of their children being so sick and not getting the help that they needed. And then foundations were established by these patients who realized that something important needed to be done, and the only way to convince doctors was really to fund good science. A lot of the scientists were afraid to accept money from these foundations because they feared that their research would be controlled, or in some ways they would be denigrated by accepting that money. The good news is that, in fact, those foundations, by and large, have let the clinicians, the researchers, be free to publish the results as they came out, to do the best quality science using research that had been peer-reviewed, and then just let it show what it shows. And that's really a great credit to all the different foundations and donors who have supported the research, including the Global Lyme Alliance and the Cohen Foundation and the Bay Area Lyme Foundation recently as well. So what have we been doing at the Lyme Center at Columbia? I'm a psychiatrist, as I think many of you know, so the first thing we did was to look at the frequency of psychiatric disorders since symptom onset, and to our great surprise, we found that in a survey, patients who had a history of Lyme disease reported depression 3 times more often than patients with non-Lyme arthritis, and then patients who had lupus. So that was certainly intriguing. Then there was a lot of concern about the fact that these are subjectively reported symptoms: fatigue, cognitive problems, depression, all subjective reports. So is there any way of actually objectively finding objective parameters to correlate with those subjective symptoms? So we started looking at brain imaging, and we did a very NIH-supported study, really nice study, of patients with chronic cognitive problems. And what we found was that they did have many areas of decreased metabolism, mainly in the parietal and the temporal areas of their brain. And we also looked at blood flow as well in the brain, and that was decreased also, but in a similar pattern to what you saw with metabolism. So basically what it showed is that there is something going on in the brains of these patients that's different from patients from healthy controls. And then we did our study, which is this bar over here. The last on the right is patients treated with ceftriaxone. 64% of them benefited with repeated IV antibiotic therapy. Everybody in our study had already had IV antibiotic therapy. So here was a second or third round compared to placebo. This was on fatigue, which was a secondary outcome measure, not the primary. What was interesting about this was that it was identical to what Lauren Krupp found in her study, and fatigue was one of her primary outcomes. So our study, which came after hers, supported her study. Also interesting in her study, embedded in one of her tables, was that patients who were IgG Western blot positive in the serum at the entrance of her study were much more likely to respond to ceftriaxone than those who were given placebo, suggesting that that might be a biomarker, at least among these patients with chronic fatigue, identifying those more likely to respond than those who were Western blot negative. Now, with Steve Schutzer and Pacific Northwest National Laboratories, as he talked about earlier, there was a study he did in which he compared the spinal fluid of patients with chronic Lyme cognitive problems to patients with chronic fatigue syndrome and healthy controls, and finding, lo and behold, that there were 692 unique proteins in the Lyme patients, not present in the chronic fatigue group, 738 unique in the chronic fatigue group, and there were, I can't read that, about 724 unique proteins in the normals. So suggesting that maybe the normals had something that the others needed, and that the Lyme patients were indeed different from the chronic fatigue patients. So that was one of the first proteomic studies, I think, done of patients with Lyme disease. Now, as we've thought about Lyme disease and the chronic symptoms over the years, certainly we think about persistent infection as one possible cause. We think about immune dysregulation as another possible cause. We think about unrecognized co-infections. But we also are thinking quite strongly now about neural network dysregulation, which is that the brain is changed as a result of the prior Lyme infection, causing an activation of perhaps the pain pathways so that they're hyperactivated, they're not turning off properly, and that repeated antibiotic therapy may no longer be able to change that because it's not due to persistent infection, but it's due to the altered neural networks. So that's one of the areas that we're studying right now. And then about 4 years ago, we started collecting, well, actually, when we were doing our Lyme encephalopathy study, we collected samples, and that's proven very helpful. And we're also collecting samples now as well, patients with erythema migrans, patients with post-treatment Lyme syndrome, patients with neurologic Lyme, as well as a group that is not very well studied, which are those patients who have an acute onset, who were previously well, but who turn IgG positive, but they don't have the objective markers of Lyme disease. They're very sick. They should be studied as well. So we're collecting samples from them also. We're doing a neural modulation treatment of patients with chronic pain. We're doing a meditation and yoga study for patients with chronic pain and fatigue after Lyme disease. With Cohen Foundation support, we're doing a pathology study along with Tim Salati and Andrew Dwork here in the corner, looking to see, there's been reports in the literature, and some of you may be aware of it, suggesting that spirochetes may be found in post-mortem brain tissue of individuals with dementia after Lyme disease, or even in patients with dementia who have no history of Lyme disease, not necessarily Lyme spirochetes, but other types of spirochetes as well. And so we're doing two different studies. One is to look at post-mortem tissue from people who have died and generously donated their brain and spinal cord to us and other tissues. But also looking at using the New York Brain Bank at Columbia, comparing people with dementia who don't have dementia to see if there can be evidence seen of spirochetes present. Shannon Delaney, where's Shannon? Raise your hand, Shannon. Shannon's over here. Yes, she's doing a really nice study looking at patients with young adults and children with psychosis. She's mainly interested in collecting a repository to see what can be learned about immune markers and infectious markers. And one of the things she's looking at as well, and specifically, is Lyme markers. But that's going to be a very interesting study. We're looking to see whether the patients with post-treatment Lyme pain have these elevated neural pain pathways. And we're doing that very cruelly by putting patients in an MRI scan and then administering thermal pain to their skin, and then then their brain lights up, the pain pathways light up, and we're comparing the Lyme patients to the healthy controls with the hypothesis that the Lyme patients would show far greater activation using the same amount of thermal pain stimulus. So it's a really interesting study, which I suspect will be positive. And then we're treating these patients with these glutamate modulating agents and then testing them again as well to show, hopefully, not only does their pain improve, but also their neural network activation diminishes for pain. So that'll be really nice if we can show that that happens. We're also using MR spectroscopy to look at the chemical components of what might change before and after treatment. Finally, it's generally believed that it's really not interesting to know whether someone has persistent intrathecal antibody production in the spinal fluid, because it really doesn't tell you anything, because it's really just an evidence of the prior infection. which may well be true in the vast majority of patients. What we've started to look at is, can the evidence of intrathecal antibody production among those who have it compared to those who don't tell us anything either about the clinical symptoms or about the neural network activation? And really interestingly, if you include all three parameters together, if you include neural network activation, Bieberdorfer-specific immune response in the CSF, and clinical syndrome, all of a sudden you start to see different patterns. But it's only if you include all three in an interacting way. And if you look at these two correlation matrices, the one on the left are the C6 positive patients, and these are clinical symptoms, psychological symptoms, neurocognitive symptoms, somatic symptoms among different patients. And similarly here, it's cutting across. So all you need to notice is that the C6 antibody index positive patients differ in their correlation color pattern compared to the C6 AI negative patients. It's a small sample size of 10 versus 15, but nevertheless, it does suggest that there is a difference in the relationship between clinical symptoms among those who are C6AI positive or C6AI negative, suggesting that there's something about that marker in the spinal fluid that has clinical meaning. And that's what we're doing right now, is trying to figure out exactly what that clinical meaning is. So what's the path ahead? Well, there's tons of paths, and we've heard tons of fascinating paths here today. So I just threw this out at the last minute. Certainly we need we need excellent and rich and full biorepositories. And John obviously has done a terrific job with his repository, and as has the Bay Area Lyme Foundation. But I just want to emphasize we do need a richer repository of patients with post-treatment Lyme syndrome, as well as with the disseminated Lyme cases. Because I think it's pretty obvious to assume that patients who have disseminated infection or neurologic disease are more likely to have post-treatment Lyme syndrome than those who present early on with the rash and get treated early. That's been shown in some European studies, and it reasonably would happen here as well in the United States. Obviously, we need a test of active infection. When we get that would be very helpful. But clinical judgment will never fail. I mean, good clinical judgment is needed in ordering any test. We need treatment studies, we need prospectively collected samples, and then finally, I think we have to start looking at what is the impact of antibiotics? What is the impact of taking many, many months of antibiotics, as some patients do, on the gut microbiome, and how does that affect the individual, and how does that affect the individual's immune response? I recently read a really interesting paper on chronic fatigue syndrome and the metabolome of chronic fatigue syndrome patients that just came out in the Proceedings of National Academy of Science. And in this discussion with, I think his name is Robert Navo, in the discussion, he was suggesting that antibiotics may have a negative effect when taken chronically on mitochondrial function. And that obviously needs to be reported more, studied more, and also looked at in our Lyme patients. And that's all I have to say. Thank you very much.
Dr. Fallon, have you included, brilliant talk, thank you. Have you included microglial activation in your PET studies? There's a nice peripheral benzodiazepine PET ligand, and I think that would explain a lot of the synaptogenesis defects.
Yeah, that would be a great study to do. There's different ways of doing PET scans. You can do PET scans just looking at the metabolism, or you could look at blood flow, as we did in our prior study. The next study, if we were to do a PET study, would be exactly that, which would be to look at microglial activation, which is the inflammatory markers in the central nervous system, using something like PK1195, which targets the microglia, and then you could do, let's say, a minocycline treatment or some other treatments with these post-treatment Lyme patients. So if it's activated, you could actually have a nice objective marker to see if that activation subsides. That would be a cool study. We actually have a phenomenal team in our neuromolecular imaging lab at Columbia, and so we are well situated to do that study. Okay, well, thank you. Yeah.
So good afternoon. So we're at mid-afternoon here, and we've spent a lot of the portion of the day talking about the immune pathophysiology of Lyme and diagnosis and treatment. So now we're going to switch gears here a little bit and move to prevention. So we're moving upstream a little bit. So looking forward to an exciting conversation. We have 3 expert panelists here. I'm going to introduce them briefly and then let them go ahead and introduce themselves. So starting immediately to my right, Dr. Rick Ostfeld is a field biologist at the Cary Institute of Ecosystem Studies in Millbrook, New York. And his work focuses on the ecology of Lyme and other tick-borne diseases. I have his book here, Lyme Disease, The Ecology of a Complex System. Fascinating read. In fact, I'd say it's not a good bedtime read because I was staying awake all night. And now we have, to his right, we have Dr. Rich Marconi. He's a microbiologist, immunologist at the Virginia Commonwealth University Medical Center. His work focuses on Lyme vaccine development, first in canines and now in humans, and his lab is focused on bringing a human Lyme vaccine to market. And last, on the far right, we have Dr. Linden Hu. He's an infectious disease specialist at Tufts. His lab uses clinical and bench research to understand host versus organism immune interactions, and he's also developing strategies to reduce Lyme by controlling infection in its wildlife vectors. So I'm going to go ahead and let them each give a brief introduction, talk about their work, and then introduce some of the important ideas that we want to discuss in this panel. We'll let them chat, and I'd encourage them to respond directly to each other, and I'll try to stay out of the discussion as much as possible, and then we'll open it up to the floor at the end for questions from the audience. So why don't we start with Dr. Hu?
Okay, great. Thank you, David. You want some reading for bedtime? I'll give you one of my grants. I thought I would just set the stage for this discussion by reviewing briefly the life cycle of Borrelia, which offers both challenges and opportunities for prevention. So I think most of you in this audience, I think almost everybody knows that, let me see. So the ticks are hatched from eggs. They hatch uninfected with Borrelia burgdorferi, and they acquire Borrelia burgdorferi during their first feed on one of the reservoir animals, and that's usually a small rodent or a bird. Those ticks, after having acquired infection, molt to the nymphal stage, and at that nymphal stage, they again feed once on rodents, small rodents and birds, and that's at the point where they complete their cycle and they pass it on to the next generation of animals. And that's really the critical cycle for maintaining disease in the wild. Humans are incidentally infected, as we've heard multiple times here, and that has advantages and disadvantages for design of therapies for prevention. And then the nymphal tick molts into the adult stage, and the adult stage tends to feed on larger mammals, deer being the classic one, but these larger mammals don't participate in the cycle. Now, in terms of prevention, I'm going to give away the punchline right off the bat, and that is that nothing, and you guys probably have noticed this already, that nothing we have tried, and there's been lots of things tried, have really slowed the advance of Lyme disease, either geographically or in absolute human case numbers. And lots of different things have been tried. So, and we'll talk about this, I think, as we go through this, but people have tried acaricides, spraying things that kill ticks. People have tried local applications of acaricides onto, either small rodents or onto deer, and that hasn't worked. People have tried shooting the deer, and that hasn't worked. Well, actually, I'll take that back. And I guess this kind of gets to the issue of precision medicine and how it might apply here. So here, if you think of the environment as the patient instead of the actual humans, I think one of the reasons where you see a lot of differences in studies that work in some places but not in others are due to microenvironments and differences in the quote-unquote environmental patient, so that one approach doesn't fit all in terms of trying to prevent disease if you try and take an ecological approach. So those are some of the challenges we face. I'll just take one more second and just talk about a project that we have going on. We try several different things in terms of prevention, but this is one that's funded by the Cohen Foundation, so I thought I'd talk about it for just a brief second. And we do a project on reservoir-targeted vaccination, and the idea there is that if you can decrease a carriage of the bacteria in one of their reservoirs, in this case we're targeting mice to start out with, then you can potentially decrease the carriage of the infection in ticks and then subsequently decrease transmission to humans. And so we have a virally vectored vaccine that works orally in a single dose. We've been testing it in simulated environments in a BSL-3. that costs us $80,000 a month to run. And then we're doing, with the Cohen Foundation support, we're doing pilot trials of baiting strategies out on an island in Massachusetts. We're currently just waiting for USDA approval so that we can put the baits out on the island containing the actual vaccine. So with that, I'll pass it off to Rick.
Thank you. So my name is Rich Marconi, and I'm at Virginia Commonwealth University. And what I'd like to do just over the course of the next few slides is tell you about a technology that we have developed that is designed to lead to the development of new vaccines for Lyme disease. We've been fortunate to have this successfully applied so far in a canine vaccine, but of course the goal now is to take it into a human vaccine. So again, the goal of what I'm going to do is highlight to you why I think we need to take new approaches to vaccine development and just very briefly outline how we're going about doing that. So I want to start by looking at a very simple experiment that we have done. And I first want to draw your attention to this rather beautiful molecule down here. So this is outer surface protein C, and we're looking at the atomic structure of this protein. And one of the we've known for some time that Ospc has great potential as a vaccine candidate and potentially as a diagnostic candidate. But in order to develop sort of the next generation vaccine or diagnostic, one of the things we really need to know is specifically which parts of this protein are actually triggering antibody responses during an infection. So what are really the antigenic domains of this protein? And again, if you look at this molecule, Ospc is not quite this beautiful in nature. This is color-coded for our purposes. So the red indicates the highly conserved domains of the protein, i.e. the amino acids that don't vary amongst all the known Ospc sequences. And then the blue and purple indicates the parts of the protein that form what we call the variable shell of the molecule. So again, is it the variable part of this protein that stimulates antibody responses during infection, or is it the conserved domain? And if you know that, that provides you tremendous insight as to how you're going to develop a vaccine or diagnostic assay. So here's the simple experiment that helped us address this question. So what we did is, as many of you may know, there are distinct Ospc types that exist in nature. There's maybe 30 or so worldwide. We generated recombinant protein for every single one of those Ospc types. And for the purpose of this slide, I'm just labeling them as, giving them numerical designations, Ospc types 1 through 6. And here we are looking at a series of immunoblots. And all these immunoblots for this representative experiment have all 6 of the Ospc proteins spotted onto them. What we did is we screened these immunoblot strips from serum from canines that were Lyme disease positive using a C6-based test and clearly infected with the Lyme spirochetes. And what I want to draw your attention to is the stark difference in the immune response to Ospc that develops during an infection. So if you look at dog number 1, for example, this animal clearly has antibody to Ospc as evidenced by the dots that appear for the type 3 Ospc and then the type 6. But remarkably, the antibody does not cross-react at all with the other Ospc types that are spotted on this blot. And if you look at some of the other individual animals that were assessed, we see exactly the same type of result. So once again, the immune response in an animal is clearly dependent upon what type of strain they get infected with and what Ospc type that animal is producing. Again, looking at dog number 6, for example, here we see a clear antibody response to type 1, but virtually no cross-reactivity with the other Ospc types. So what does this tell us? Well, what it tells us is that it's not the red part of this protein that's triggering antibody responses, because if it was, the antibody in an infected animal would react with every Ospc variant. That's clearly not what we see. So it must be the variable shell that triggers antibody responses. So what we did is we wanted to ask specifically, well, is it this part of the molecule? Is it this part? What exact domain of the variable shell triggers antibody? So to do that, we did some epitope mapping analyses. And I won't go through those studies, but basically the outcome of those studies were that we identified 2 linear epitopes in the Ospc protein. And having identified those proteins, we reasoned that we could utilize them to develop a new generation Lyme disease vaccine. So to highlight this goal, what we did is, let's pretend these are different ticks. It's actually the same tick rotated different ways. But if you take these different ticks and you recover the Lyme spirochetes from them and you look at their Ospc type, you'll see based upon the color coding that there's a lot of variation among them. So we reasoned that if we took the 2 individual epitopes that we identified and pieced them together along with the same epitopes from multiple Ospc variants, we could generate a brand new protein that's not Ospc, but instead is a protein consisting only of the productive parts of the Ospc molecule. And we call these Chimeritopes, which stands for a novel protein that consists of protective linear epitopes derived from multiple Ospc variants from multiple Ospc strains. So I'll end on that, and I'll just tell you that what really excites us about this technology is the ability to now develop a very broadly protective vaccine, but also to incorporate into these Chimeritopes epitopes derived from other proteins, whether it be other OSP proteins or potentially proteins from Anaplasma or Babesia or any other pathogen that you choose, with the goal being to develop a multi-pathogen vaccine for tick-borne diseases as opposed to a simple Lyme disease vaccine. So with that, I thank you, and I'll pass it over to Rick.
So thank you. Thanks for sticking around so long and paying attention as well as you have. I've been learning a great deal about precision medicine, which is something that I have not been familiar with up until this morning, or at least not very familiar with. And I've come to understand, among other things, I still have much more to learn, that precision medicine takes as its point of entry patients who are already sick or who are likely to be sick, and in this case, with Lyme disease. And the point of precision medicine is to do a better job, as I understand it, with treatment and diagnosis with some attention, as we've seen in the past few minutes, with vaccines as well. That's unquestionably important, but what I want to do is provide a broader context for those patients who are already sick. And the context starts with the fact that, as we've heard earlier today, there are various co-circulating pathogens together with Lyme disease for which precision for Lyme disease diagnosis and treatment, that might not convey many advantages for these other co-circulating pathogens. A step back from that, we know that all of these are transmitted exclusively or nearly exclusively by the bite of the black-legged tick, at least in North America. And then beyond that, we know that the abundance of infected black-legged ticks, the creatures that are out there trying to make you sick, is influenced by a whole host of different ecological effects, ecological conventions, and I list those right here. So my question to all of you is, do we really want to cede all this territory, upstream territory from the sick patient, to this tick enemy, if you'll permit me a militaristic metaphor here, or do we want to actually reduce the number of people who are getting sick in the first place and who need the precision medicine that is geared at better diagnosing and treating them if indeed they're sick? And I would argue, obviously, we do not want to cede all that upstream territory. We actually want to do a better job of reducing the probability that you or I or our loved ones will be bitten by an infected tick. So to that end, with funding from the Stephen and Alexandra Cohen Foundation, we have embarked this past spring on what we call the Tick Project, fancy name. The subtitle is Testing Environmental Interventions to Prevent Lyme and Other Tick-Borne Tick-Borne Diseases in Our Communities. The co-leader of this with me is Felicia Keesing. And briefly, very briefly, our study design consists of imposing 2 different anti-tick interventions. Those are this bait box right here, which is a small box. It attracts small rodents, which, as we've heard, are the reservoir hosts responsible for infecting ticks with Lyme disease, Babesia, Anaplasma, Powassan virus, and the like. The small rodents walk into those boxes. They self-apply fipronil, which is an acaricide that kills all the ticks on those particularly important hosts. That reduces the number of ticks and, most importantly, the number of infected ticks. This is a commercial product that has been shown to be fairly successful in small-scale trials. The other intervention is a fungus that's infecting that engorged tick right there. This is fungus called Metarhizium anisoplii. It is a native fungus to forest floors, forest soils of the eastern United States. In fact, it's quite widespread. It attacks and kills arthropods of various kinds. We're using a commercial strain called MET52, which has been shown to have fairly high specificity for ticks. Our basic design is that we are imposing these interventions, either one, both, or a control, so neither, on entire neighborhoods, where a neighborhood consists of approximately 100 adjacent properties in a real neighborhood. So we have 6 replicate neighborhoods that we are imposing both bait boxes and the fungus. We have 6 replicate neighborhoods where we're imposing the bait boxes but an inactive fungus, so this is a placebo control, we're just spraying water. We have 6 replicates where we have inactive bait boxes, so the bait boxes are out there, but they have no fipronil in them, they have no tick-killing agent. And then we have 6 replicates that are controls for both. And indeed, these are real controls. These are placebo controls in both cases. So what we're doing at the level, our test tubes are entire neighborhoods, 100 homes, a few thousand individuals in the entire study. So what we're trying to do at this huge scale is to impose a scientific gold standard, which is a randomized, placebo-controlled, double-blind study. And let me just finish up by telling you what it is we're following up over the next 4 years with these interventions. The main response variable are per capita cases of tick-borne disease within the human residents of those households. We also want to know the number of encounters that people and their dogs and cats have with ticks. And lastly, we'll be pursuing the abundance and infection prevalence of ticks that are crawling around in the yards on those properties that we're treating or not treating in this experimental design.
Okay, thank you. So, well, Linden already alluded to it, or really stated it outright, that none of these ecological interventions to date have proven effective as far as reducing the incidence of tick-borne disease. And Rick, since you gave the war analogy, we'll go with that. And it seems like we have a whole arsenal at our disposal. Why haven't any of these weapons proven effective? And I'll start with you.
Well, yeah, this is an unfortunate situation because we've had various different weapons in our arsenal to continue. I'm not liking this analogy very much, but there are a lot of. We know how to kill ticks. We've known how to kill ticks for a while. To a large degree, there's been inadequate funding to do a study that's of a scale large enough to ask whether there's an epidemiologic consequence of killing ticks. To some degree, we don't know where in the environment we should target. We don't know how big an area we should target, and we don't know how strongly we need to reduce the tick population to give rise to an actual health protection response. So without being too long-winded, I could go on and on, but that's the basic idea. So the beauty of having support from the Cohen Foundation is that this kind of research falls between the cracks. NIH is not that interested in environmental interventions. The National Science Foundation is not interested in human health. That's not their purview. So this kind of research, how do you control ticks in the environment sufficiently well to actually protect human health, falls between the funding cracks, as it were. So someone needs to step in and provide the funding to do this right, and we hope that we're making an attempt to do so.
So it seems like, in our discussion before this conference, a paper came up, the CDC paper, I think it was Hinckley from earlier this year. This was a 2-year randomized controlled trial of a caricide pesticide aimed at reducing the tick population. And maybe I would ask you to discuss in a little bit more detail, but the study failed. And so, as we seek to increase funding for this kind of research, it seems that we're not doing a good job by showing that these interventions are effective. Can maybe, Linden, do you want to comment on that study and why, or I can throw it open to any of you.
I can comment a little bit. Failure is such an ugly word. It had a lot of unexplained things. So the study was basically peridomestic spraying of acaricides and killing ticks around people's homes. And the study showed that it was pretty effective at getting rid of ticks. They were way down, not zero, but way down. But that didn't have an effect on cases of human Lyme disease. And there are a number of different possibilities for why that might have occurred. And I would say, again, just to go back, this is not the first time we've seen these types of results that are a little bit discordant and hard to explain. For example, with the bait boxes, where you're applying acaricides directly to the mice, we've seen them be very effective on, say, an island site in a trial, but then not effective on a mainland site. Or with deer killing, it was very effective on an island site where you could wipe out every single deer by helicopter shooting. But in a suburban site, it wasn't as effective. So I think it gets back to the idea that, in different ecologies, and different ecologies can exist, 100 yards from each other. I think Rick is better to speak for this than I am, because I'm a doctor. I never wanted to get involved in this. I want to deal with the patient side. But very different ecologies in terms of tick transmission, what the ticks feed on, what the reservoir hosts are even. What are the important reservoir hosts in a specific micro ecological environment are very different, and that can really affect how these things work. And that's where I think I absolutely agree, much more research is needed to really understand the impacts of these different variables that will then allow us to target. We do have a fair number of things at our disposal in terms of an arsenal, and how to best deploy them and where to deploy which ones, I think, is what we really need to figure out.
Could I just follow up? So that was a very eloquent response, but I just want to add a little bit more information to the CDC study. So they sprayed bifenthrin to individual backyards, front yards, backyards, and they had several thousand properties in three, in 3 different states in the Northeast. They were successful in reducing questing tick populations, so those are the ones that are crawling around in the environment, by about 60%. So that's reasonably effective in reducing the abundance of ticks. But they had no impact, these sprays, with the placebo compared to the placebo controls, on either the number of ticks people found on them or cases of tick-borne disease. So there are probably many possible reasons why that experiment failed to protect the public health. One is that 60% reduction in tick populations is inadequate. You have to do a better job. It's one of the reasons why we designed our study as intensively as we have, and with 2 interventions in combination rather than 1. The other is that maybe people are encountering the tick that made them sick, not in their own yard, but in their neighbor's yard or down the street somewhere, or the local dog park in their neighborhood. And so that led to the idea that we need to treat entire neighborhoods if we're going to be more successful than that particular study was.
Okay.
And I think an important point you made there was that I think everybody who's looked at this really thinks that it's not going to be just a single intervention that works, that even in a single environment, you probably do need to have multiple different approaches at the same time.
So I do want to get to discussion of vaccine component as well. So maybe I'll pose this next question to Rich. Your vaccine work is fascinating, and it seems like there has been some successful vaccine work done to date, but it hasn't been successfully deployed. What would it take? What would you need, or what do we need to do to get a working vaccine deployed so that we can reduce the exposures to Lyme disease?
Sure. I mean, that is certainly a real challenge. I think that one thing that I believe has really hurt the development of a human vaccine for Lyme disease was the experience with the LymeRix vaccine. And there are many reasons why LymeRix failed, of course. Some are a little less clear than others, but there were clearly also some pretty clear-cut scientific issues that resulted from the use or the development of an Ospc-only vaccine. So I think as we move towards developing vaccines that consist of what I would call more appropriate antigens, we're going to find that the vaccines are going to be more efficacious, and that's important. And certainly as we see the spread of Lyme disease increasing, it's going to increase interest in getting a vaccine into the human market. But the big challenge is bringing in the partners who are going to take it forward for full development, no doubt about it. Industry, Big Pharma, has been frankly scared off by what we saw with the LymeRix vaccine. So the goal is really to try and sort of re-educate not only the public so that they are not wary about a human Lyme disease vaccine, but also in many ways re-educate industry about how we can take new approaches that won't have the same complications that we saw with LymeRix.
You told us to talk amongst ourselves,
yes,
right? So,
please,
so
go ahead.
can I ask Rich a question? So, when the LymeRix vaccine was around, one of the concerns that I had, and I think many people had, was that if people are vaccinated and they feel safe from exposure to Lyme disease, is it possible that they might relax their vigilance against tick bites? And because we know that these same ticks transmit other pathogens, that they could then become more likely to get infected with Babesia or Anaplasma. And if this is a realistic concern in the realm of epidemiology, would we want to then tailor educational materials to go along with vaccine delivery so that people don't just think that now I don't have to worry about ticks?
No, that's a great point, and that's something that gets addressed a lot in veterinary medicine. And I do a lot of talks with veterinarians discussing this issue. And the one thing that we always make clear is that in spite of what we believe is a very effective veterinary vaccine, you can't stop flea and tick control. That's critical. But, you know, again, as I alluded to, I think the goal is to shoot for the sky here, and that is we do not envision a future vaccine that's simply a Lyme disease vaccine. What we want to see is we want to take the technology that we've developed and make a single protein vaccine that can actually protect against multiple tick-borne pathogens. And I think we have, you know, we've proven that this general approach can work, and I think we can make that happen. Now, having said that, I think it's also important to note that Lyme disease is clearly the leading tick-borne problem. So it's hard to see us backing away from developing a vaccine for something that is a real clear-cut human health concern because of the concern that perhaps people will pay a little bit less attention to other diseases. So, we have to make sure that people don't forget about these other diseases, but let's just take it a step further and make a vaccine that works against these other diseases.
Actually, can I ask you a question too? And it's going to be one that we were talking about in the back of the room, so I think you're ready for this.
Okay.
But I think it's an important discussion to have. So, you know, I think one of the reasons that has Big Pharma a little bit gun-shy about this is the potential market for the vaccine. And part of that, when Ospc came out, was, you know, that the recommendations from CDC were very narrow in terms of who should get the vaccine. Do you think that with the increasing numbers of human cases and with the increasing geographic risk that those recommendations should change? Or do you think that they still should be the same recommendations that were in place for when, even with a more effective vaccine, when LymeRix came out?
I think absolutely those recommendations need to change. And it's really remarkable. Again, I come out of the microbiology human side of medicine, and it's only in recent years that I started interacting on the veterinary side of things. And it's unfortunate that we don't talk a little bit more, because one thing that veterinary medicine does that we do not do is they have remarkable epidemiological data that maps the spread and the risk of Lyme disease in canines. And that's where, you know, this test called the SNAP4Dx, which is used routinely by veterinarians anywhere close to a potential endemic region, tells us how this disease is spreading. So if you look at the prevalence maps put out by the Companion Animal Parasitic Council, it's remarkable to see where Lyme disease is going. And a look at that map easily tells us that we need to, you know, we need to make it clear that that same risk is spreading to the same degree in humans. So CDC would certainly play a huge part in achieving that goal of that awareness.
Okay. Well, I'm going to open it up in just a moment to the audience. So if you do have questions, just raise your hand and the runners will come to you with a microphone. But I just want to go ahead and ask one last question. So, you know, this panel is called, or rather this conference is Lyme Disease in the Era of Precision Medicine. So I do want to bring in precision medicine. In the morning, you know, the education panel talked about precision education. Can we talk about precision prevention? Is there a role for big data? And if you could collect any data, what would you collect? I'll open that up to anyone to answer.
I'll make a quick comment on that, just very briefly. I think one of the things that where we have really lagged behind in terms of developing diagnostic assays as well as in developing vaccines is we've taken an approach that is not fine-tooth enough, in the sense that if you look at whether Ospc, for example, really is a good marker for early infection, the answer would be no, unless you happen to get infected by the strain that produces the same Ospc type that's on your immunoblot. So instead of looking at things in terms of are we picking the right antigens, I think we have to move towards are we picking the right epitopes? And that's a critical difference, I think, compared to what we have been doing.
Okay.
Let me take a shot as well, and this is following up on what Linden said from the very get-go, is I think there are two areas in which at least this sort of big data aspect of precision medicine might be useful in prevention, in all this upstream prevention. One is the notion that these ecological contexts can dictate to some degree what kinds of interventions are likely to succeed and what are likely to fail. This is a big data question where, you know, the watchword of this meeting so far has been, so far as I can tell, garbage in, garbage out. The necessity for non-garbage big data in the ecological realm is essential if we're going to pursue that pathway. And that's a tough one because funding for ecological studies on tick-borne disease is virtually non-existent. The other, though, is human behavior. So we really know next to nothing about what are people doing and where are they when they encounter that tick that makes them sick. And so I think one possibility, we were talking about this earlier this morning, that if we had real-time data on people, you know, with their various devices, whether it's a cell phone or some wearable thing, that if you're willing to give information, and you give it all to Google anyway, so give information to the scientists about where you are at any given moment of the day. And then if you're prompted, you know, every 10 minutes or however frequently you can stand, maybe only a few times a day, to do a quick check to see if you have a tick on you, we could probably gather mountains of information that could be geo-referenced, so we know where you are, what's the habitat right there, what were you doing, were you with your dog or not, and to be able to better understand where it is we're encountering that tick that makes us sick. That would be a possibility that would be, I think, more towards the precision medicine type framework and could be extremely helpful. But it would be expensive.
I don't have too much to add to that. I guess I have a couple of examples. One gets back to the immunology group, and that is that there clearly is data that certain people respond differently to vaccines. So that becomes important information as we're designing new vaccines because certain people may respond better than others. The other kind of falls along Rick's last comment, and that most of the things that we recommend for prevention have to do with tick avoidance, and either pulling ticks off or showering or things like that. And again, in studies where they've done trials, they look pretty good to the extent that trials exist. But then in real life, where you tell people these things and let them go, they don't work. And there may be data around that predicting who responds to that, and also what kinds of reminders will help people adhere to those types of preventative measures.
Okay. So we have about 5 minutes left, so I'll open it up. Dr. Lewis?
Yes, so I understand that attempts to get rid of ticks so far have not been successful, and I appreciate that maybe one day it will be. But I was wondering about a different approach to eliminate, let's say, a particular pathogen like Borrelia burgdorferi, for which you could bait supposedly with an antibiotic. And in that case, you don't need to get rid of all infected ticks. Then the maths of epidemiology come into place, the herd principle, where you eliminate only a certain percentage such that transmission becomes ineffective. And that may be a more effective strategy. I wonder if that has been done as an experiment.
Do you want to take it first? Or you want, so there's been a lot of modeling around that in terms of the transmission threshold, below which you fall, below the where the reservoir rate of infection is low enough that you don't continue transmission. So there have been a lot of studies along those lines. In terms of eliminating individual pathogens, I mean, that is the approach we're taking with the reservoir-targeted vaccination, with the idea that perhaps you damage the environment less by just targeting a specific pathogen rather than spraying acaricides, which may have other side effects. But antibiotics have also been tried. So doxycycline has been looked at and was actually pretty effective. I think the concerns with doxycycline administration, this was a study done by Nord Zeidner, was that it's the only agent we have right now for anaplasma. If you develop resistance, and it's one of the more clinically useful drugs for Lyme. And if you lose that agent due to resistance, and I know you're an expert on resistance, so you can appreciate that, then that would be a disaster for human health.
So what we have is an ecological problem, and it needs an ecological solution. Lyme disease was discovered about 40 years ago, and it was in a small section of our country at that point in time. And now it is among 46% of all the counties in the United States of America. As Dr. Ostfeld said, the problem has been that there hasn't been enough funding for people like himself in that field doing large enough studies. And I have spoken with these researchers because from the get-go, 20 years now, I believe that we have to have an environmental solution that has to deal with the ticks. And if we don't deal with the ticks, there is going to be continued spread of ticks, continued increase of the other diseases, as we've been seeing has been happening. And if you treat just Lyme disease with a vaccine, it's just a patch on the problem. We've got to deal with the problem. And I assure you, Dr. Hugh, if you spoke to the scientists in the field, they will tell you how poorly funded they have been, way less than what the rest of you get. And Lyme disease isn't funded enough, Lyme and tick-borne disease. Dr. Beard showed a chart at our integrated tick pest management conference in DC that showed that there is 10 times as much reported tick-borne disease as mosquito-borne disease. And then if you apply the 10x, that's 100 times. Yet I'm sure the funding is nowhere in comparison. The funding needs to be done. It needs to be given to people like Dr. Ostfeld who can do something with the source of the disease. I mean, even the CDC, Nucatone is a great thing.
Sure.
They could develop a soap that they could wash themselves with afterwards when you come inside and reduce tick-borne disease. There was an anti-tick
Yeah, if I could
vaccine.
comment on that real quick. Just to clear up one notion, I would venture that we would all agree we're not overflowing with money in any way whatsoever. But I do thank the Cohen Foundation for the support that we've been given. But there's one point I'd like to make. I agree that this is obviously an ecological a problem of ecological origins, but we should not lose sight of the fact that many of us are sitting in this room today because of the effective development of vaccines against infectious diseases. So vaccines have proven time and time again to be perhaps the most inexpensive and powerful approach that we have to preventing infectious disease.
But a vaccine for an anti-tick vaccine that would prevent them from staying attached to people so they couldn't transmit any diseases whatsoever would be effective.
Okay, thank you.
Yeah, and I would say that, yeah, that would be great. And I know there are multiple groups working on it. Steve Weichel's worked on it. Tom Mather's worked on it. We've taken some of their antigens and looked at it. Unfortunately, I think, again, it gets back to some of the dynamics of the tick and the feeding that we haven't been able to find an antigen that's very, very effective. The best we can see is about 50%. And so I think we need to do a little, it's not that it's terrible, and it might be part of a combined vaccine, but I think it's not enough alone.
And it's not just the antigen, it's also the fact that for a vaccine of that type to be fully effective, you have to constantly maintain high circulating antibody titers in vaccinated individuals, which is a daunting task that requires frequent boosters, and that's generally something that the public has not proven particularly receptive to.
Yeah, I have a couple of questions. I want to first thank you for the work that you've been doing. I live in an endemic area in Massachusetts called Dover, and I wonder if you guys have thought about pheromones. I notice that on my street, every household, people have been bitten, but some people can be out organic gardening and they never get bitten, whereas I won't even step on the grass anymore because of my experience. And I also worry about what's in the blood supply because we're not testing for Babesia. I worry about sexual transmission. I know that hasn't come up today. It hasn't been proven yet, but there's a lot of theory behind that. And then lastly, well, there's two things. Having taught in the Czech Republic twice where the kids are vaccinated and they feel very protected, I also wondered about what Watson did with fruit flies so many years ago. So those were my 4 questions. Sorry, they're all in conseculars, but there you go.
I'm not quite certain about the pheromone question. ticks do use pheromones. There is a mating pheromone that they use to aggregate, and then apparently there are pheromones that cause them to cease activity, which is hard to tell from a tick not ceasing activity. But that seemed connected to the question about whether some people are more attractive to ticks than others. It wouldn't be human pheromones that influence their attractiveness to ticks, because pheromones communicate within a particular species. And there is interest in using pheromones to either enhance other tick control agents like acaricides, or to try to immobilize ticks or get them to move into the wrong place at the wrong time so that they're not that likely to attack us. I think this is worth pursuing, but it's also important to realize, I was serious when I said it's hard to tell an active from an inactive tick. I mean, it would take a tick the entire day to crawl across 2 sections of this table. So if you're putting a pheromone out over here, you've made that tick move, a meter or 2, and so their ability to influence their behavior in a meaningful way is somewhat limited. The other questions I'm going to let my colleagues address.
That's one thing. I literally observed my daughter on a yellow towel on Martha's Vineyard in the hot sand, middle of July, 20 feet away from the grass, and I saw 3 ticks moving on an angle towards her.
Okay, those were probably Lone Star ticks. So.
No, these were deer ticks, and if I hadn't seen it, and if the people that I was with hadn't seen it, never would have believed it.
Collect
So
them.
unfortunately, we're going to have to cut it off
I'd love to
here,
look at them.
and we can continue the
Can I
discussion
give one quick answer
afterwards.
to that?
Yeah, sure, go ahead. Yeah, go
So
ahead.
don't take this for any, but somebody mentioned that we do a xenodiagnosis study. So we're getting information on tick feeding from this, because these are all laboratory reared ticks that are from the same source. And we're definitely seeing differences in patient feeding. So there are people who we put these ticks on, we put them on repeatedly, not a single tick will take a bite. Where there are other ones where we put the ticks on and they feed repeatedly very, very well. We don't know what it is. I think Sam Telford, who's the person who works with us, has some theories about garlic and B1, but I don't think those are validated. So I throw that out there. Maybe I'll ask Rick if he knows anything about that. But there definitely are differences in people. I don't know if it's pheromones. I don't know what it is. Some people are tasty. Some people aren't.
So I want to remind everyone to stick around. We have the closing keynote to come and the reception afterwards with booze. Let's give a round of applause to the panelists.
Thank you, everyone.
Thank you.
All right, so just to keep things moving along, we're going to get started here. So we have a short talk about LymeKB, which is a component of this Cohen Lyme Foundation Consortia that centers on what we're doing here at Mount Sinai, and Brian's going to talk about it. But as an introduction to this, we had a lot of discussions today about big data, a lot of people leveraging new tools like genomics, proteomics, et cetera, to leverage big data, and talks about how we can bring this into the fold of Lyme's disease and Lyme precision medicine. So one of the things that we've been tasked with is bringing this to Lyme disease, right? So bringing the power of big data. We've been applying it in many other diseases like cancer, like Alzheimer's, and other areas. And one thing I want you to think about when Brian's talking is, how can we help you guys, right? So you guys, by you folks, I mean the amazing men and women we've heard from today that have been working in the trenches here of Lyme disease and have all kinds of valuable data sets. And we've talked about sharing data here and that being very empowering for Lyme and many other diseases. But to really think as you're going through this, how could someone like us, how could the types of resources we're building really help you with the burning questions that you are all walking around with, and with the data sets that you guys already have in hand or are generating. So with that, I'm just going to have Brian come up here and give an introduction to the concept behind Lyme Knowledge Base with an early glimpse into some of the stuff we're doing there. But really the purpose here is to generate some discussion and on how we can build these types of resources that really serve the needs of this community.
Thanks, Joel. So I'm going to, like Joel said, give kind of a motivation behind how we're applying big data, where we applied it in the past, and then show you some of the new developments that we've had around building this knowledge base for Lyme. So some of you probably are quite aware, The digital universe is vast. We're at 4.4 trillion gigabytes as of 2013. Most of this is coming with cat videos on YouTube, but there's also a number of biomedical data that's contributing to this. So thousands of gigabytes are now dedicated to biomedical data. And one of the things that we've talked about and sort of ties everything together is we're trying to really tap into this vast repository of data, whether it's collecting data on the environment, collecting data from whether that be your smartphone or other things, and pulling that all together with the sort of more traditional, whether it be molecular profiling or clinical data from hospitals or other outreach clinics to build better models of disease. So to ingest all this data and to really dial down to better diagnostics and better therapeutics. And that is in both the individual case as well as for population health. And, there's been, with these kind of new tools and technologies, scientists are extremely good at drilling down, and we have the ability to have high resolution and even breadth on the data that we can collect, the measurements that we make. How many of you, this I throw up there, and this is what scientists might see, and if we, this would be like an individual investigator, their expertise, looking at this picture, and that's, they're really good in their domain. They have detailed knowledge about whatever they're studying. But what we'd like to do is take a zoom out and collect everything to see the broader picture. So collectively, we're all working towards, all these individual scientists, physicians, and whoever are working towards this bigger picture, right? This larger canvas. And we're all making these contributions, and we're trying to tap into that. And that's really what this big data and systems approach is about, is how do we actually pool all of our resources to be smarter than any one of us individually for the good of patients and for better understanding and treating disease. I just want to come back to this real quick because this was an important point that I kind of glossed over. So this was the case history of this kid who had a fever of unknown origin. It persisted for about a year, and after the third hospitalization, they finally agreed to try next generation sequencing. What wasn't really shown in that was the timeline for that. So next generation sequencing, this whole approach took place over 2 days, right? So this is, for those of you who are actually in the trenches doing next generation sequencing, that's really fast. And this was an urgent case that needed, all hands on deck. And what's fascinating about this in particular is the actual analysis time is about 2 to 3% of the total time. It was this last 90 minutes, at the end of day 2, where everyone's, finishing up and saying, oh, here's what we found. And this was, spirochete, this is a leptospirosis bacteria that they actually decided to go ahead with. But it was on this kind of hunch from unbiased sequencing in a 2-day blitz to try to figure out what's going on with this patient. And so what has happened, we have this really, we're in this golden age of a lot of data from scientists are now made publicly available. One of the main repositories for publishing data, making your findings known, is something by the National Library of Medicine called PubMed, where these are all the scientific studies are indexed. And what's shown here is over time, over the last 50, 60 years, this has grown exponentially to now we're at 2015, 2016, we have more than 26 million articles across every known biomedical investigation possible. And in some small fraction of that is targeted toward Lyme disease. And we can use this as a reference point to actually learn what is all the knowledge in the biomedical sciences on Lyme disease. And in addition, we now have these fantastic tools. So in the mid to late 90s, a company called Affymetrix started this revolution where they went from a single gene measurement to making tens of thousands of genes measurements at one time. So now individual investigators have in their hand the ability to query tens of thousands of genes in a single shot. And what this paved the way for was you could do, and in addition to this, investigators, one of the prerequisites of publishing this data is the journal said you have to make the data available. It was the Wild West in the late 90s and early 2000s, and people were getting all kinds of conflicting studies reporting that this gene was important or that gene was important, and it wasn't until everyone settled down and said, okay, let's see the data, let's make it available, where you had this sharing mechanism for the data and allowed better analysis tools, statistical tests on those data to see what was actually a real driver of disease versus not. And over this process of early 2000s to now 2016, this one repository, and this is just on gene expression information, has grown exponentially to where there's now 2 million samples in the public domain that any high school kid, anyone in the world, my grandmother, could download this data and start playing around with it, start asking questions about it, because it's now freely available and in a fairly standardized form so that you can make these comparisons. And this is actually what, when you have this at your disposal, there was an inflection point around 2008 where you had enough of these studies in aggregate to start asking smart questions like, what does the entire diseasome look like? How do we compare studies across all these different diseases? What do we find that's similar? What do we find that's different? And this is one of the things that Joel actually pioneered, was taking these molecular profiles and saying, hey, gee, we have all these molecular profiles of disease. Can we actually use those, combine those with drug profiles, and ask, this is the match.com for drugs and diseases. So you take a drug profile that's supposed to it perturbs a cell, it changes its it changes gene expression pattern. You go through this massive public repository of molecular profiles and ask, what's the match? In this particular case, what's the match that would be the best treatment? And Joel really pioneered this and showed this was possible to go across a bunch of diseases. And since then, the lab has gone on to look at options for repurposing this in inflammatory bowel disease, in various cancers. And then more recently, we looked at actually targeting specific immune cells. So going out into this vast complexity that we have with individual immune cells and asking what drugs may actually start to manipulate specific immune cells, either activate or inactivate them in particular ways. And so it's all made possible by the fact that this data are publicly available and they're at large enough scale to where we can ask statistical questions about what's valid and what's not. And so that's on the sort of data molecular matching side. In the sort of patient phenotypic space, what we want to do is shift towards, we have all these clinical information, but that's usually housed in repositories that are not accessible. At Mount Sinai, we have one of the largest electronic medical records. We have about 6 million people in the electronic medical records. We are the largest healthcare system in New York. And so we asked, what can we learn from this electronic data that would allow us to better subcategorize patients? And in this recent paper out of the lab, we were able to actually identify subgroups of type 2 diabetes strictly based on their lab measurements and sort of their clinical phenotype that you would see from the data in the electronic medical records. And then we went on to actually validate this in another electronic medical records at Columbia. And so this really allowed, this was the first glimpse at starting to use large data to actually subgroup patients into these various islands. And we have clinical categories for them, but then recently we went further and applied some deep learning approaches, which are just machine learning algorithms on steroids to go through all of the electronic medical records and actually start predicting what patients will develop simply based on their lab values and how they're categorized from a disease perspective in your clinical history. Okay? And so that's on the subgrouping, providing better precision medicine for fine-tuning. And then more, sort of the holy grail of problems is how do we learn from the text-based information that is part of the scientific literature. And so one of our recent recruits, Beth Perchef out of Stanford, actually has done a lot of work to parse out all the text from what's been published to derive meaningful relationships that allow us to say, okay, this molecule upregulates this cell type, or this molecule is active in this disease. And so it opens up a broader set of data that is not necessarily transcriptional or genetic in nature from one of the public repositories, but is how humans think about and describe their scientific findings and talk about the relationships between either molecules, chemicals, and disease. Okay? And so from that, we're at this point where we can ingest all this information and start to learn the broader patterns and then really dive down into what would be better ways to diagnose and treat patients. And so how have we been going about this? Well, we've already said that Lyme is a complex disease. There are many, whether that's ecologically, immunologically, or even sort of host response and treatment responses. And so what we're trying to do is build up this, embrace this complexity and take in all these sources of data on individuals. And from that, we've been able to generate reasonable models in other systems that have led us to make better therapeutic or even offer diagnostic options. And we're hoping to do that in Lyme. And how are we doing that? Well, part of that is thinking about the data space and the structure of that data. So there's the public data that I just mentioned too, which is the scientific literature and what has been either by fiat or just by community agreement to publish your data, whether it be genetic, proteomic, other cytomic, and so forth. And that's readily accessible, and we've pulled that down. The other, I guess, that we're inviting people and really excited to work with people on is this more unstructured data. So this is the stuff that resides in lab notebooks. Healthcare records, for good reason, are kept private and sort of segregated off from everything else. But thinking about ways to actually get access to de-identified data that is tied back to some of the other measurements, because that really is where the value is. It's difficult if you only have molecular data to say something about patient outcomes if you don't have those outcomes and what the patient actually, the patient's history and so forth. And then there's, all this sort of either clinical trial data from vaccines or drug treatments that is also not necessarily easily accessible. So with that, we decided to create something that we're calling part of the first step of this was to take a survey of the landscape. What exists already? How do we bring that in on Lyme? And what are sort of the structures and features of LymeKB? Well, first of all, we created an open source tool. What does that mean? Well, it means that it's not necessarily proprietary software. Anyone can download it. Anyone can use it. Anyone can contribute to it. It's actually designed to explore across not just Lyme, but make connections to other diseases, link up with what potential treatments are because of disease-disease similarity or gene-gene similarity and so forth. The other is this, the model that we're putting forward actually is empowering to actually run statistical tests, right? So these are models that make predictions that would allow you then to validate those predictions and see, are they true, are they not? That's sort of the hallmark of science, is making a predictable model that leads to some validation that will either disprove your hypothesis or invalidate your assumptions. The last is, part of this open source initiative is we would love it if other people contribute their tools, their software to make this better, custom to whatever analyses they'd like to do. And we have a set of analyses that we do. Other people have analyses that might be more, clinically focused, might be more proteomic focused, might be something else, ecologically, environmentally. So this would allow for the sharing of those kind of information and those tools. And the sort of the model that we're basing on, we have all these cloud computing tools. All of this resides currently in the cloud. If you don't know what that means, it's just we took Amazon servers, and all you have to think about is you can go to your computer and pull up the website, and everything's available. Right? So it's always on and always out there, and it's just through the login you can get this information on your desktop, on your laptop, whatever. And so just as I've just pulled down, it's always risky to run live demos, so I've just pulled down a few screenshots of this, but what the hell. So this is one of the, this is sort of the Simply front end. So this is the natural language processing parser that takes all the information on Lyme disease and what the other term is. Is it co-occurring with another disease? Is it co-occurring with another gene? Is it co-occurring with a specific chemical? And from that we can infer what the relationships are, whether they're meaningful, gene-gene connections, possible gene treatment connections. The other aspect, so the other one is we pulled together all the molecular and whether it be genetic data, where it existed, proteomic data. So I would actually thank Bill and Mark for a lot of the proteomic data. This is from the literature, plus information from various tissues that allow us to actually say, okay, this gene is active. So for example, the classic TLR2, that's highly relevant for Lyme disease. And researchers can click on whether or not there are particular drugs that would target TLRs. What is the evidence, so you can actually go to the primary sources. What are the papers that cite that TLR2 is important for Lyme? So a lot of biologists and scientists don't sort of believe the big data. They want to go back to the primary reference, read the paper. So we have those links as well. So this is the start of building this. It's small only because at the moment the available data is small. It will hopefully grow with everyone's support and contribution. So just to bring this back to, this really is, we're hoping is a team effort. So, it's been great. Our collaborations have started with Johns Hopkins. They're collecting a lot of really rich data, clinical, that there's, Johns Hopkins sort of seems to be the hub where they're sending their samples around to everyone. So to Kim Lewis's group, to Monica's group, and doing all this profiling that then we're going to bring back together into what we're hoping will be the first actually multi-scale model of Lyme disease that really pulls together all of these parameters on individuals, right? And will allow for better diagnostics and better therapeutic options. And with that, I'll just thank all the people who have been involved and take any questions if there are.
To connect graduate students that are biologists with the tools that you're developing and the threshold that exists between experimentalists that like to work on the bench and sitting on the computer pulling down this data. So how do you, collaboration is one thing, but at the end of the day, if you have a graduate student, they want to look at their own data. They want to be creating this data, and whether it's primary data or data generated by looking at databases. So we need the training tools, I think, to generate this next generation of graduate biologists that are comfortable looking at this data and not leave it sort of in the hands of the few. And it seems to be there are a few hubs right now that are using this data very effectively. Your group is one of them. So that it's just really disseminated and not sort of everything funnels down to Stanford or Montana or whatever. So are you having graduate training programs available, or how are you thinking of doing this?
Yeah, so I completely agree. It's a great comment. And one of the things that we're going to offer are training sessions. We haven't set the sort of pacing for whether those are going to be, how many times a year they will be, but that would be to actually really bring together the computer scientists and the biologists for all sort of life sciences, because that mix has really proven very effective, because the biologists have really great questions. Computer scientists are lousy at the questions. They're really good at looking through the data. If you kind of tell them this is, these are the kinds of questions that are important, go. But yeah, we definitely have a lot of sort of outreach and educational components planned that would be around training for sure. Yeah. And so, yeah, I guess this is a call to if you have interested graduate students, postdocs who are coming from the life sciences, please have them get in touch with us, because we'd love to bring together that mix. Great.
So we have the final talk of the day. If we can locate him. Where's David? Oh, there he is. So it's David Roth, and I think one thing we felt was important was to end the conversation with a perspective of a patient, right? So we've had a lot of talks today about data, about molecules, about genes and proteins and bugs. And I think it's really important to go back and anchor all this in, before we leave today, to remember why we're all here and the perspective of a patient. So I'm really grateful to have David here. Took time out of an extremely busy schedule here, and I think has a really unique perspective, because not only is he someone who's affected by Lyme, but is someone who also decided, outside of the, I don't know, 80 hours he probably works a day at his main day job to try to do something about it and establish, foundations to help other people. So I think you offer a really unique perspective on how everything's been unfolding in this field and where the opportunities lie ahead. So thanks.
Thank you. Good evening. My name is David Roth. I'm a senior managing director in the real estate group at Blackstone here in New York. I run Latin America for us, so I'm traveling down south all the time. First of all, I want to thank Mount Sinai, Joel Dudley, for holding this event and for inviting me to talk to you about my experience. I also know a lot about the amazing work being done by many of you through the funding of the Stephen Alex Cohen Foundation. I don't know if I saw Alex here somewhere, but I know Alex and Jeannie and Chris were here, and I just want to thank them for all they're doing. I've been involved, as I'll discuss, for the last 5 or 6 years, and I think what this effort is truly one of the most, it's truly the most, in my opinion, exciting, promising, and hopeful things that's happening within the Lyme world today. I also want to thank all of you who, through some strange combination of curiosity and other factors that only motivate researchers, you've all decided that researching tick-borne disease is worthwhile and have somehow found yourself here tonight. So why am I here? I was diagnosed by a CDC positive blood test for Lyme disease and then a clinical diagnosis of Babesia in August of 2010, 4 months after becoming ill. In the search for meaning and the hope that I could fight what I saw as a sort of a social injustice that was occurring as a result of the challenges faced by many suffering from Lyme and other tick-borne diseases, I ended up forming a nonprofit called the Tick-Borne Disease Alliance. Ultimately, this group became the largest charity focused on tick-borne illness in the country. I've spent much of my life since becoming ill focusing on the myriad issues that affect sufferers of these diseases, both current and future. And I'm pleased to see many people here whom I've met and gotten to know through my work in the area, including my friends from Bay Area Lyme, with whom I worked on the National Lyme Biorepository. My purpose tonight is to provide a few minutes of the perspective of the patient population. And though I really haven't been asked to do this and maybe have no business doing this, provide a few thoughts that relate to the business you're all in, or many of you are in, research and solving problems. A bit of my story. Like most chronic sufferers of these illnesses, when I became ill, I didn't see a Lyme rash. I didn't know what symptoms to look for. I went to doctors who didn't know the symptoms of tick-borne disease and who relied upon faulty diagnostic tests. I had 2 negative blood tests before a CDC positive test 4 months after becoming ill. I was treated on and off for about a year with antibiotics, anti-malarials for the Babesia, and a variety of other drugs, supplements, dietary changes, and frankly, anything else I could try that might help, from detoxifying foot pads to faith healers. When you're chronically ill, you will do whatever you think might help. I spent unbearable amounts of money, probably in excess of $100,000, all the time being lucky enough to afford whatever treatment I might seek out, and countless hours of pain and suffering that went beyond the pain of the illnesses themselves. This is a very common thing with the patient population. I struggled convincing doctors to take me seriously because, like many sufferers, I didn't quote unquote look sick, and my blood work was inconclusive and confusing. After 2.5 years of illness, I finally reached the level of wellness where I currently sit. I'm about 85% of the human I was before I got ill, and continue to suffer periodic bouts of my original symptoms. I now speak to upwards of 5 or 6 sufferers a week, or people who have children who are suffering, and the stories are remarkably similar to mine. Unexplained illnesses that are difficult to diagnose, and a result of non- or misdiagnosis become difficult to treat. Unlike me, most aren't lucky enough to have unlimited funds to seek treatment. They rely on an insurance industry that has been unwilling to recognize the plight of these patients, largely because our understanding of the disease is limited to what a handful of historic experts have said about these diseases. Who knows whether my ongoing symptoms are caused by my initial infection and the havoc it's wreaked on my immune system, or ongoing persistent infection. That question, I believe, remains unanswered as of yet, as does the issue of whether a year of antibiotics helped me or forever destroyed the internal flora needed for a proper working immune system. But until we have a better understanding of the pathogens found in ticks, better diagnostic tests that are sensitive and specific, and precision therapeutics that truly cure these illnesses in a way that doesn't bludgeon the patient as the current antibiotic approach does, people will continue to experience what I did. Symptoms and a medical community that seem to plunge the patient into Alice's rabbit hole at a time when the patient is most vulnerable and most in need. Some statistics we've all heard before, I think, are worth repeating as they reflect the magnitude of the problem, and I apologize, I wasn't here, so if you've heard this 7 times today, I apologize. I'll focus on Lyme without, trying to minimize the impact of the myriad other tick-borne illnesses that the medical community has seemed to be able to discover at what seems to be a rate of several new diseases every year. As we all know, the CDC recently provided an analysis that the number of annual infections of Lyme alone is in excess of 300,000 per year, growing at the annual compounded historical growth rate from 1994 to 2013 of about 6%. We'll potentially witness a growth in infection rates over the next 5 years to over 500,000 new cases per year. And that's assuming global climate change doesn't increase the rate of growth of Lyme and other vector-borne diseases. And it also doesn't take into account the strict restrictive nature of the surveillance definition. Research by John Alcott, who I see is out here, has shown that even if caught quickly, upwards of 20% of infected Lyme patients remain ill after the typical 3 to 4 weeks of antibiotic treatment. So pretty soon we'll have at least 100,000 people a year who will go on to experience extended bouts of illness. The cost to individuals and society is massive, by many accounts, several $billion per year being spent on diagnosis and treatment. And this is just the United States. In short, there's a huge market for all you researchers and for the biotech companies and venture capitalists to chase. There's a reason that I describe it as a marketplace, because a few reasons. One, where we are with government funding requires that people from outside of the government fund. So that's a combination of high net worth individuals like the Cohens, but also the marketplace. And there truly is a market for the diagnostics and therapeutics that all of you can develop. But it's also, I talk about it as a marketplace because it's important, I think, to remember that just having technology that works is not enough. For a diagnostic test or a therapeutic to really change the lives of sufferers, it needs to be cost-effective, mass reproducible, in addition to being technologically capable of doing what it's supposed to do. And for therapeutics, obviously, they need to do more good than harm. And it all needs to be backed up by hard data showing its accuracy. So in the spirit of trying to get you out to the alcohol as quickly as possible, I just want to leave you with a few thoughts. The first relates to the act of doing research that you guys are involved with, which by definition tends to require narrow focus on well-defined cohorts. My request is that you remember the patient experience. Remember the patient experience when you are designing your research, as well as, and more perhaps as importantly, when you are drawing your conclusions. By way of example, the issue of development of new diagnostic tests. By focusing just on Lyme disease and ignoring the complexity caused by other Borrelia as well as non-Borrelial tick-borne infections, diagnosticians often lower the odds that we will be treated appropriately and that we as patients will get well. Consider the challenge for the test makers. There's the obvious weakness of any serological test in that they're inaccurate during the 3 to 4 weeks before antibodies can be measured. But beyond that first month or so, it's often claimed these tests are fairly accurate. Unfortunately, for the patient population, that belies the fact that many, many people become ill despite being seronegative, according to the ELISA, the ELISA and the Western blot tests. Now, you guys use hypotheses all the time. One hypothesis is that none of these people have Lyme disease. Well, we know that at least some portion, and I would guess a fairly large number, might be seronegative because of exposure to antibiotics early in the illness. We also know that some might be suffering from something like Borrelia miyamotoi, which is a spirochetal pathogen that comes from the same ticks that transmit Lyme disease and has now been identified as a source of human illness. Borrelia miyamotoi is estimated to have caused illness in several thousand people in the U.S. Annually, and it's a bacteria carried by ticks, causes Lyme disease-like symptoms, and is treated with antibiotics, which sounds and feels a lot to the patient like Lyme disease, yet doesn't result in a positive blood test. In this context, the hypothesis that none of the seronegative patients have Lyme disease just seems too simplistic and lacking in nuance, which gets to the main point I want to make. You as researchers often draw conclusions from well-defined cohorts that are often very small in sample size and only representative of the broader world up to a point. In Lyme disease in particular, this has been the rule. As you develop your diagnostic tests and therapeutics, I implore you to think about what it must be like to be a patient. Consider carefully whether it is appropriate to make the conclusions you derive from very small, well-defined cohorts and apply them without reservation to the broader, more complicated population. This has been going on for 30 years within the community, where we have a very small number of samples that have defined how the medical community applies both diagnostic criteria as well as therapeutics. This idea of Lyme disease and the era of precision medicine is a terrific one because it seems like there's no less precise area of medicine than this, in my opinion. My second point, and I just would call it a hope, is that you all will consider the patient experience as you consider how to interact with your colleagues. While siloization has been a problem in many areas of science, I believe the political aspects of Lyme have made the tendency towards this siloization even worse. Only by open discussion in fora such as this, and by providing data to big data providers like the Next Generation Healthcare Institute, if I'm getting that name right, will we be able to make the rapid progress that current future sufferers need. So thank you for having me, and thank you for being here. And happy to answer any questions.
There's a threat targeting America, one that's growing fast, but may still be hard to see. Lyme disease. Spread by tiny ticks, often smaller than the head of a pin, this dangerous disease is now more widespread than West Nile, tuberculosis, and HIV/AIDS combined. And if left untreated, it can lead to arthritis, facial paralysis, and even memory and concentration problems, often called brain fog. As the threat of Lyme disease grows to more than 300,000 projected cases each year, it's time for us to target Lyme disease. That means checking for ticks when you've been outside, even in your own backyard. It means seeing a doctor if you experience the symptoms of Lyme disease, which can include joint pain, flu-like symptoms, fever, fatigue, or sometimes a bullseye-shaped rash. Set your sights on stopping Lyme. Learn how you can target Lyme disease at targetlyme.org.
I just want to briefly thank everyone who's just especially the hardcore here folks who stuck around all day. It was a long day. It was an exciting day. I want to thank all the speakers who took valuable time out of their schedules to come and spend a day with us. And before everybody goes and gets a drink, I want to thank the awesome staff here who helped us. Savi Glow, who's sitting right here, Melissa Nieves from our team, and Freddie Reyes, and Dave Stark, and Greg Stock, and Brian Kidd, and I'm sure I'm missing someone else, Franco, and others. So this was awesome. So, all right, so please, and Kathy Stetcher, yeah, thank you. So please go out and enjoy some food and drink. You've earned it.